Wastewater is complicated stuff. There's a lot of stuff in it. There's a lot of biological material, there's a lot of non-biological material. There are a lot of microorganisms in wastewater, and there are pathogens like the SARS-CoV-2 virus. Dr. Carrie Schultes pushed for the efforts on our campus to do wastewater testing.
When the pandemic hit, universities everywhere were trying to figure out how to stay open, right? And how to keep students in their buildings. That conversation led into what other economical ways can you test a lot of people? And the natural answer to that is wastewater testing.
Last semester, I started working with Carrie, and I was doing the data analysis along with one of the statisticians in the math department. What it allows us to do is to use the wastewater samples as a leading indicator to identify cases of COVID to prevent outbreaks on campus and keep our campus open.
So initially we sent all of those samples to a molecular analysis lab in Fort Collins, and they were really expensive samples. And so the university was invested in trying to find another solution to bring that sampling in-house so it wouldn't be so expensive. What we did was we went after a federal mineral lease grant, and we were able to buy this really powerful, most high-tech piece of equipment on the market, digital PCR machine. You know, this piece of equipment doesn't exist between Salt Lake and Denver. And so we set up this whole molecular analysis lab, and so now we can do all of that testing here on campus for a fraction of that initial cost. So everybody in engineering really had to work together to create a sampler inside of a larger system that could answer the questions that we're trying to ask. How many people in these dorms are sick? So the student sampling team works together to coordinate their schedules. There are 8 samplers total, and so we're doing stuff most days. The only day we're really not doing something is Saturday. So we have a big jug, a 5-gallon jug, where the sample is ultimately housed. We have a pump, we have a tube that connects to the pump and goes down into the sewer, and then we have a battery. And so each day that we're gonna sample, and we sample 3 times a week, we deploy our sampler, which means we set the sampler up to start to take the sample. 24 hours later, we go get our approximately 4 gallons of wastewater that came straight out of the sewer that's well mixed from the residents of the dorm that it's serving. And then we take a little vial, 40 mils, out of our big 4-gallon jug, and then we take that over to the biology lab so they can tell us the concentration inside that 40 mils.
By being able to do things on campus, we cut the time between wastewater collection and SARS-CoV-2 quantification by approximately 24 hours. The sooner we know where people are who might be infected, the sooner we might be able to step in a way that limits spread. When it came time to do wastewater testing in-house on this campus this spring, I, along with my colleague Dr. Zeynep Azoy, were asked to put together a lab that could do this work and to develop the procedures that we would use to detect the presence of SARS-CoV-2 in wastewater.
There are several labs who are doing it, and they've been very generous to share their protocols with us, but none of them were doing it exactly the way that we were going to do it because of the equipment we had. Basically, I wrote up the protocol that will be used in our lab. The lab requires a person who's there the full time. The faculty, like myself, we have to teach. We cannot really go in and do 2 hours of work and then go teach and then come back, do another hour of work. Once you start it, you have to finish it. So we needed someone as a technician to work in the lab.
My name is Anton Carey. I graduated last semester from CMU with a degree in biology, and I am currently running the wastewater lab. Dr. McQuaid, at the end of last semester, asked me, would you be interested in this position? I can't guarantee it, but it's a possibility. And so I, just graduated. I'm like, I'll take a job. I'm in charge of doing the actual processing itself, from sample concentration to dPCR amplification that we do in this room. We get wastewater samples that are obtained by our engineering students, and what we do is we concentrate the virus that's in there and we get it separated and mostly on its own, and then we process it so that we can get just the RNA, and ultimately we bring it in here so that we can amplify it and see how much was in the original sample.
One of the things that's really important about setting up this capacity to detect virus wherever it occurs and then to sequence it right away is we can identify if there's anything going on of concern or any new lineages. We can do all sorts of investigation. The peacock variant is essentially this combination of the California strain that's emerging with this new mutation that also seems to be rising. And we saw it kind of become very quickly very common in the viral samples that we're getting from CMU. That could be chance, but it also could be something else, and we want to pay close attention.
I think our ability to do this quickly and effectively is really a testament to many parties at CMU to kind of roll their sleeves up and do the hard work that is detecting the presence of SARS-CoV-2 in wastewater.
I feel like we had a goal. We had, the time ticking. Make this get to work as soon as possible. I'm also very proud of our students who are working. They have really stepped up.
It's been really invigorating to get to work on something that really matters. We have been deeply mission-driven to make sure that we can stay in person.
The work that we've put in, we've been able to identify a few cases and prevent some outbreaks. It's humbling.
The whole process has really given me an insight into perseverance that's necessary in science. Every experiment wasn't, perfect and giving us the result we wanted, but we just had to keep on trying it again, changing the procedure to make it better and more efficient, and ultimately get to somewhere where we can get readable results.
The most rewarding part of my job is to train students in doing science. And I very much like that in this effort, we are able to successfully engage students in the art of doing science in a way that will give them a better appreciation of what science is, how it's done, what it can do, and how it might affect the way that they think about the career paths that they might ultimately follow.